I am Ariely Palanca, presenting the bioremediation of contaminated groundwater to remove nitrate, chlorate, and perchlorate using zero-valent iron or ZVI and emulsified vegetable oil or EVO. Aside Nevada has been found to be contaminated with high concentrations of dissolved anions such as the nitrate, chlorate, and perchlorate. These dissolved anions, or what we call contaminants, impact the human health. Luckily, these contaminants are potentially biodegradable, and bioremediation has been suggested as a cost-effective treatment method. To observe bioremediation, a carbon source must be available to bacteria to reduce the contaminants to harmless compounds such as chloride and nitrogen gas. Currently, the groundwater of the site has been treated by an ex situ bioremediation. This is a process that requires pumping of water out of the ground and treating this water in fluidized bed bioreactors, which is costly. An alternative approach is to treat the groundwater by an in situ bioremediation. This does not require the pumping of water. Literature research and previous studies performed at UNLV have demonstrated that contaminants present in the groundwater are potentially biodegradable, and several carbon sources can effectively support the biological reduction of nitrate, chlorate, and perchlorate. However, ZVI, which can provide electrons for the reduction of these contaminants, has not been investigated. Our hypotheses for this research are ZVI may perform, as well as other electron donors, for the reduction of contaminants of concern, and naturally occurring bacteria in zones are sufficient to promote degradation by both ZVI and EVO. With this, our research aims to evaluate the use of in situ bioremediation to remove contaminants from groundwater and to determine the potential degradation of contaminants using ZVI and EVO. Our research method is to have two bioremediation columns that can be used to mimic the well and its flow at the site. The picture on figure 2 shows two columns that were tested during this research. The column on the right has sand, while the column on the left has a mix of sand and ZVI. These columns were fed with the groundwater containing EVO and diammonium phosphate or DAP. It can be seen on the table influent feed matrix the concentration of EVO and DAP that were being fed on the columns. EVO has 400 mg COD per liter of concentration and DAP has 5 mg P per liter. Below the table, the setup for the bioremediation columns can be seen. The entire column is 2 feet tall, where one column has 378 grams of sand, while the other has 264 grams of sand mixed with 113 grams of ZVI. The influent feed was pumped in the column with a downflow and constant flow rate. Then, the effluents were sampled and tested for pH, nitrate, chlorate, perchlorate, and chromium. This research experiment was done for 43 days, and the three graphs on the results show the level of degradation that took place. The empty bed contact time, or EBCT, is the time where the treated groundwater was in contact with the treatment medium. The column with sand and CVI has 105 hours, while the column with sand only, the one without CVI, has 11 hours. The graph for the nitrate degradation shows that the one with CVI has completely degraded, while the one without CVI only degraded for the first half and stopped degrading for the last half. This happened when the influent concentration had changed to a higher concentration. The graph for the chlorate shows that the chlorate on both effluents had been highly reduced throughout the experiment, and the graph for the perchlorate shows that there is degradation only on the effluent with ZVI. The effluent without ZVI did not degrade at all. After looking at the results, it can be concluded that an in situ bioremediation can treat contaminated groundwater. There is potential degradation of the nitrate, chlorate, and perchlorate with the use of the ZVI and EVO. Also, there are naturally occurring bacteria in the soil that help degrade the contaminants. And lastly, as shown in the results for the column with sand and ZVI, the bioremediation column test is more effective with the use of ZVI in reducing nitrate, chlorate, and especially perchlorate. Thank you for listening.